All right, welcome back. Here we are on Chapter 7. We're going to be talking about biotechnology, and it's a pretty interesting topic. And being consistent with trying to serve my students, I have a sort of an overview of careers that are in biotechnology. You can go to that link, or you can grab my slide set and just click on this slide. It'll take you there. But it lists various job possibilities in the field of biotechnology, and you can find out about those. They tell you how much you can earn, and various things and what it takes to do these various careers. I just wanted to make available, these are hands-on. As you learn things during this course, I hope it opens to vistas. When I was starting out, I didn't know really what I wanted to do, and the best thing to do is to kick tires and to find out what interests you, because a lot of the things I didn't know were available at the time. And as I got in uh, deeper and deeper into my career, my college career anyhow, then it really started to gel for me as to what I wanted to do. Now, uh, Wake Tech does offer uh, a variety of non-degree short classes. We started a new Associates in Science program. It won't be offered, I think, until 2021, which is right around the corner. But currently, we have these courses, and these are overviews and various things that you can do. Some of these can be found at the Biotech Center that's on NC State's campus. Now, you don't have to be an NC State student to take some of those courses, and we offer them in conjunction. So you might need to go and travel to the Biotech Center there. It's something if you're interested, you want to go visit, because it's an amazing facility. They have a two-story biofermenter that's there that you could see, and people working. It's alive. It's actually working. It's making at least the last time I knew, it was green fluorescent protein for research purposes. Anyhow, these are interesting. You can get these certificates and get these under your hat. It helps you in your career. So I just wanted to bring that up. You can hit the link for this down at the bottom, or just click on the slide. And the same here. There's a little video there that you can play. Again, you can just click if you're interested in a career there. So as far as our goals for my presentation and for this particular chapter 7, I just want to kind of give you an overall tour of the basic tools and how biotechnology is applied. Um, some examples of how we can produce various different types of pharmaceuticals, treating disease and things using these biotechnology tools. And then try to give you just a brief overview of genetic engineering, gene jockey, recombinant DNA technology, PCR, and transgenic organisms. And that's going to be huge in your career as you move forward. These will be things that will be going on. And of course, gene therapy, and there's strides being done now in that area. It's amazing. So biotechnology, I like to view it as an agent for change. I was lucky enough, I guess, to be my career started about the time it was exploding. Biotechnology and genetic engineering were brand new, and I was just at the time in school, so I was had majored in genetics, or actually minored in genetics and majored in microbiology, and using you scratch a microbiologist, we bleed genetics. Uh, Microbiology was used, the microorganisms were used to understand the DNA technology areas. And so I got a really good look at that. At the same time, the computer technology was starting to explode, and this was in the late 70s, early 80s. And so the two kind of overlapped each other. We utilized computer technology to help us with the biotechnology tools, and it was quite an amazing time to be a student learning these things. In agriculture, there's tremendous number of examples of how biotechnology is used, crop resistance to pests and various things. Human health, treating disease, there's new advances being added all the time. Forensic science, again, way beyond just the basic fingerprint, DNA fingerprinting, using PCR technologies and being able to recover from a crime scene small amounts of DNA, amplifying that now and then determining who was there. If you're at a scene, it's going to be really hard not to leave some evidence behind. That's kind of how it is these days. So, using biotechnology, first of all, what is it? Uh, it's a very broad term. Of course, bio just meaning anything related to life. The use of technology to modify organism cells and their molecules to achieve practical benefits. That's a pretty broad definition. It's a pretty good one. Included in this overall header, genetic engineering, you will find, is of course the manipulation of the genetic material by adding, deleting, and or just basically cutting and pasting genes from one organism to another. Is quite amazing what we're doing, and I'll try to introduce you to that. But this is actually, it's been in progress for quite a while. There was a lot of promise. I remember in the early 80s, it was the promise that we're going to have all these things. Well, we're actually seeing that now. It took a while, but like anything else, it takes time to have new changes. I'll give you an example of each one of these as we go along. It won't be directly in order, but that's okay. We're using bioprinting. You know, 3D printing is near and dear to my heart. Using 3D printing technology to print organs, they're actually doing this at Duke, at Chapel Hill, UNC at Chapel Hill, various others. There's quite a few places actually, but you're basically placing certain cells that will grow, and you can place those strategically to either recreate an ear. There was a big Nature article about that. A mouse was growing a human tissue ear, and we can print it. And so we're making advances. It's not totally there yet, but it's well on its way. And the hopes are that biotechnology will bring this technology where it'll save lives and that sort of thing. GMO, genetically modified organisms. I dare say there's not any food that you eat, with the exception of maybe two or three that I can think of, that aren't already modified genetically. Here's some that are corn. Almost guaranteed 99% of the corn that you get to will be GMO. Sugar beets, soybeans, potatoes, apples. A lot of these engineerings are to improve shelf life and other characteristics. And in third world countries, sometimes they're adding some nutrients through genetic engineering that part of the world may not be able to have access to. So there's a lot of things, and there's good and bad associated with GMO. Some feel that GMO products can cause negative side effects to health, and we'll talk about that a little bit later on. I'll have a paper for you to review about GMO, and then you render if you're for or against GMO, and I'll be posting that soon. And so be looking for that. I always post an announcement whenever I make a change. But a GMO is, of course, under the biotechnology label, and in the forensic science having access. Now, when I mentioned the merging of technologies back in the 1980s, when we saw the biotechnology sort of ramping up and the computer technology ramping at the same time, it was two independent explosions that intermingled very nicely technology-wise. And so with forensic science, one of the big boons, of course, is automation. And the Human Genome Project, we'll talk about that. But leveraging multiple computers and automation. In other words, you don't need a lab tech to physically do all the chemistry and manipulations. You can do that via an automated system. And there's some a team that was developed that bid and got the contract to do the Human Genome Project. One of an individual, Dr. Craig Venter. If you get a chance, if you're interested in startups and that sort of thing, Craig Venter's written a book about several books. He's an amazing man. He's done some incredible things, but one of them was the Human Genome Project, and he was a master at using computer technology and in the laboratory setting and expediting the redundant process of DNA sequencing. And that's sort of what you're seeing here is machines that all they do is DNA sequencing. And I kind of show you a slide here. So when they do the sequencing in the computer, what they try to do is to slowly find out what nucleotide is at a position, and one by one. And when it does, it assigns the C's and the G's and the T's and the A's a certain color, and then those now can be read by a computer to tell you exactly what the sequence is, and then we'll list those. Not only that, it can compare it to other existing sequences and show how they differ or what their maybe origins are, and all sorts of uses for this. But it used to be, now I've done actually DNA sequencing the old way back when I worked at Bird's Wellcome, and I was a graduate student there at the same time. It's a great way to go through school because you see applied, and also I was fortunate enough to land a grant to pay for my stipend while I was working at Bird's Wellcome. And I sequenced some genes associated with virulent bacteria. And doing that was a chemical process and the maximum Gilbert technique and all this. But the idea was, I said, gee, this is a really important skill that I need, and it was complicated and all that. And I could do probably 100 base pairs. It took me about 2 weeks. And there's 2 strands in DNA, so I had to do both strands just to kind of, if it validates my work, to see if I sequence properly, and reading the gels and all those sorts of things. Well, computer automation, look at the graphic here. This is representing a sequence, a very long sequence, and it's all automated, and all the color bands and the various things are read by the computer to generate the code. And so they can rerun it, but it's literally a matter of pushing a button and it goes through the chemistry to generate the sequence. And this is important to you because now we can do tens of thousands of bases through these processes in a day. And so that means that eventually you're going to be able to go up somewhere and yank out a hair, and a process like PCR will amplify the DNA, and you'll get sequencing information, then spit it out and tell you, what diseases you might have or are susceptible to someday. There's a movie called Gattaca that did a really good job kind of capturing the future on that sort of thing. If you get a chance, Gattaca is quite an interesting movie. Usually in labs I show that, but I don't know a good way to do that, so I just let you know. It's for the time it was produced, it's quite a very insightful movie. It really gives you a good feedback as to what's going on now and how it will be applied in the future. And in some ways it's very scary, but in other ways it's good to understand that. But here, anyhow, this is just one aspect used in forensics. So in summary, biotechnology modifies it's a technology that we can modify organism cells and various other things for practical benefits. Now, yes, there could be used to less favorable things, cloning babies and things. We'll talk about that. It's been done. Primary areas in which biotechnology is applied include agriculture, human health, and forensic science. It's just the tip of the iceberg, really, what's going on. So, 7.2, we're going to talk about some of the basic technologies that sort of are the underpinnings of biotechnology. And a lot of these are just really manipulation tools, DNA manipulation tools. In other words, I can cut it. Once I cut it, I can take that piece of DNA and amplify it, and then I'll have enough copies, and then I can do additional techniques so I can cut and then insert this piece of DNA into another piece of DNA, and then I can have bacteria express that or just amplify it or whatever really I want to do. And genetic engineering is, again, this was in the 80s. I was taught that basic technology using restriction enzymes and various things to cut DNA. And we can cut it precisely. There's some uniqueness about these restriction enzymes. Is quite an interesting story. I hope I can share some of the things. I was very lucky to attend a seminar by one of the gentlemen that identified restriction enzymes. Quite an interesting man. But at some later date, I'll share some of that. It's really interesting stuff. So when I say chopping DNA and all that, the restriction enzymes are the key. These are proteins usually isolated from bacteria, like EcoR1 is a restriction enzyme. It's from E. coli, rough strain 1, and it's an enzyme that cuts DNA precisely at a sequence, and only that sequence. So if that sequence exists, this enzyme will cut it. And so that is the preciseness of the cutting. It is an enzyme that does the cutting, and what's nice about it is that we can glue or put these back together. So I can put an EcoR1 cut DNA with another EcoR1 cut DNA with DNA ligase, which is really quite straightforward. And so we can use these enzymes to become gene jockeys, I guess you could say, and insert new fragments of DNA. Now this is a plasmid out of DNA. I'm not going to have you worry about that. But the bacteria, this is extra chromosomal inheritance. You have the bacteria chromosome, and then you have these little circular pieces called plasmids that allow us to do all these things. They're naturally occurring in bacteria, and then we've engineered some of those plasmids to do what we want, and the bacteria will replicate it faithfully and express it if you want it. It's quite an amazing technology, and again, that's why I was mentioning that being a microbiologist, you bleed genetics, because the basis of genetics came from that field. So here is sort of an overview of what I was talking about with the enzyme. Now it's a protein. An enzyme is not consumed in the reaction. It's free to go and do it again, but when it recognizes or clumps onto DNA, it recognizes a particular site. Now in this case, this enzyme recognized ATCGAT. I just want to point out that it is quite interesting. It's a palindrome. So if you go on the other side, it's ATCGAT. It's the same sequence but in reverse, and that's a palindrome. And all the restriction enzymes, with exceptions of blunt ends, I'll talk about that later, a few of them are not, but for the most part 99% are, that have these sequences that are palindromes, and then cuts. Now this particular one will only cut between the A's and the T and separate these 1, 2, 3, 4 bases, and then break the phosphate backbone on this side between the A's and the T, and that will now give you what they call a sticky end fragment. And if you break apart that will have two of the ends of that restriction enzyme that now can be ligated or could be cut or put together with a plasmid that's been cut with this enzyme and put it back in, or whatever we want to do with this. It's just a matter of throwing around some enzymes and precisely in a controlled environment. Another really important technology was polymerase chain reaction. So in bacteria we can, let me show you, in bacteria we can clone, like I was telling you, the gene of interest, insert it in a plasmid, and then have the bacteria now replicate and make copies of that plasmid, and then ultimately we can get that gene back. Well, a faster technique, although this works, and it works quite well, having bacteria become your sort of manufacturing facility for whatever gene you want to clone. PCR, polymerase chain reaction, actually the gentleman that identified this really very basic technique got the Nobel Prize for this. And basically what you do is you heat DNA to a point around, I don't know, 78, 85 degrees, somewhere in there, and it shakes so violently that it overcomes the hydrogen bonding between the bases, and it separates, leaving of course the phosphate backbone intact. And separating it allows us now to get things into that region. And for right now, in order to amplify DNA, you have to have a primer. And a primer it can be synthesized in a DNA synthesizer, so we can have particular sequences that we know are there and synthesize the primer. It can bind, and then we can extend. Once it binds, we can extend. Now it provides that 3' hydroxyl group to move. Now don't worry about catching all this. All I'm telling you is that we can amplify DNA simply chemically by heating and cooling and repeating that over and over again. And so we can start with, let's say, one strand of DNA, go through the cycle where we heat it, add the primers, extend those primers out, and we can make a copy. Now we have 2 copies, repeat by heating, cooling, and alike, and now we have 4 copies. And the process can go on however many you want. And so we can get buckets of DNA if we want using this technique, and only that DNA. So this is biotechnology. This is sort of a newer tool. This is an older way of doing it, but achieves the same sort of thing. In the day, we used to cut certain genes, we didn't know what they were, and then have them, they call this shotgun cloning. I don't know if you've ever heard that before, but it's a shotgun approach. In other words, you just cut DNA up with restriction enzymes, ones that you know that you can insert into a plasmid, and then you allow the bacteria to express whatever fragments of those genes are, and then you go randomly through and your gene library and find whatever fragments that were in those bacteria, and then you can study them and all that. This was a very prominent technique, but if I go back here, and I don't mean to confuse anyone, but I was telling you that we can DNA synthesize this primer. That's the new thing. Once we have the Human Genome Project, where we've gotten the entire sequence of the human genome, and now pretty much we've sequenced so many things, we have all that information in computers, and now we can synthesize those primers and amplify whatever DNA of interest that we want. So even this technology, the shotgun cloning, has become sort of passé, although it's still used by some labs. This is still some of the older technologies. So in summary, let me go on to that. Modern molecular methods make it possible to copy DNA from one organism and deliver it to another. So we can take it from DNA is DNA, and with some exceptions of methyl groups and things, but we can take the DNA, move it around, put it in different organisms. That makes it easier to study. And the methods include the using of naturally occurring restriction enzymes, which there's a growing list. I think the last time I looked, around 78 different restriction enzymes that recognize different sequences, and they're always finding them. And they're usually from bacteria, but it doesn't have to be. It could be yeast cells and all sorts of things. For cutting DNA, We can use polymerase chain reaction to amplify DNA, and only DNA that we want. And then we can use cloning vectors or viral vectors, whatever we want, to amplify, transfer DNA. And so we can move. So the whole process of copying DNA is sort of roughly called cloning. Now, cloning has gotten some, it's really a broad topic, but cloning is just amplifying a particular region of DNA, or putting it, moving DNA from one to another. And so the question becomes, is it okay if we clone humans? And I have to tell you, it's already been done, or at least it's reportedly been done. And this starts to get into the real ethical dilemma. Now, I would invite you to go see it, or to rent the movie Gattaca if you haven't seen it. It does talk about this, genetically engineering human traits. Don't you want the best of you? You know, that sort of thing. Don't, just rely on just random processes. Let's do it genetically engineering, and forget the normal processes of procreation. This way you can have offspring that are exactly what you want. You don't like the color of eyes, well, we can change that sort of thing. Watch the movie if you get a chance, Gattaca. And so you can go look for that. There was a time where someone has cloned a pet. This was in California, if I'm not mistaken. It was a company, and they successfully cloned an animal. And then what the idea was, when Fifi died, the owner would say, "Gee, I'd love to get Fifi back." So when the new dog, was cloned and all that, and it survived, and got this cloned dog, of course the dog didn't recognize the owner, and the owner was like, "What's the point? I mean, I might as well just go out and get another dog instead of spending, $100,000 for one." So the idea was, it can look just like Fifi, but it's going to have to relearn and do all those things. And of course, the company went out of business, and the idea didn't catch on. So it first started with Dolly. I don't know if you remember hearing about that, cloning sheep, and they had done various things. And factor 9 is a really important gene. I won't go through a whole lot, but it deals with hemophiliacs and blood clotting. And factor 9 was really hard to get. There's factor 8, all different factors, blood clotting factors. And so we clone this in the genetics, and we can have it expressed and then given to some people that are missing this particular clotting factor, and save their lives and improve it. But Dolly was a sheep that was cloned, a whole sheep. So the idea of cloning your pet kind of came out from that. And then, of course, we had primates created by embryonic cell nuclear transfer, by using essentially going in with a syringe. And then there's Dolly, the one I was making reference to, the first mammal created by somatic cell nuclear cell transfer. So we cloned from this animal, took a cell, and that cell now became a new organism, and cloning that. And we've done this with primates. Stem cells, you've probably heard a lot about stem cells. Regular cell has a subset of the keys that can open various doors or turn on various genes. A stem cell has all the keys. So a cell can become any type of cell given circumstances and signals and various things. So it has all the keys and can turn on the genes. That's why stem cells, you may have heard about, and we'll talk more about those down the road. But that's all biotechnology. And of course, human embryonic stem cells were created by somatic cell and transfer, so they can work on those. And there's some ethics, because it uses the baby's placenta and various other things. And so this technology was more of a brute force. The newer technology that I wanted to bring up is technology called CRISPR. Recently, the two researchers, two women that came up with this new technique, was actually developed from bacteria, if you can believe that. In bacteria, it has a very crude immune system. It's constantly being bombarded with viruses, and the way to deal with it, the bacteria developed or evolved this very rudimentary sort of immune system, and that's the basis of what CRISPR is. It's developed from that, and they got the Nobel Prize for this. And CRISPR is clustered regularly interspaced short palindromic repeats. Am I going to ask you that? No. It the meaning of this and how this works is actually a lot easier than the name kind of implies, but it's a way of editing DNA. In other words, I can take a sequence that's got maybe some sequence pairs in it that are unfortunate, maybe mutation, and I can generate now a new sequence using DNA synthesizers and sort of exchange the malfunctioning sequence with one that is correct, and correct that issue by inserting that DNA literally in you, and then you now express the proper gene, and then off you go. Now, they've done this in mice, and as I always joke my dentist, because in mice what they did is they regenerated teeth. So teeth started to regrow using CRISPR technology in these mice. So when I go to my dentist, I said, could I get the CRISPR to regrow these teeth that you're about ready to drill, and just pull it and just regrow it? How's that? And he looked at me like, like I had 6 heads or something. But actually, we're not that far from that. So we'll see what happens. It won't be in my lifetime, but it'll be in yours. So CRISPR is a gene editing system, and after identifying a particular sequence of interest, in other words, let's say these control the signals to go on and turn on, let's say, growing a molar or something like that, and the sequences for the CRISPR RNA and Cas9 enzyme were introduced. So there's the CRISPR and then a segment called Cas9, which facilitate now the movement of the genetic material. So it sort of holds in one area in the Cas9 the gene that we want to replace, and then we can insert the new sequence by the RNA guide that sort of places that new sequence into where it needs to go, and then the DNA has the proper or the inserted sequence, and it'll be fully functional. So we can cut out the old, insert the new, using the guides, and the Cas9, which kind of is the holder for that, and the CRISPR technology is the molecular manipulation part of it, and we now can insert any gene that we want. And so gene therapy is taking on a whole new light in the future. So and I'm not going to expect you right now to grasp all of the features of CRISPR, other than the fact that we can introduce genes in vivo, in other words, in a living organism, and make those changes permanent. And examples for things that we can use for CRISPR: mosquito DNA can be altered to block malaria from spreading. Human trials are underway, and CRISPR altered white blood cells are being used to fight cancer. CRISPR technology, beagles have been engineered to be born with twice as much muscle as a typical dog, which I'm not really sure why we want that, but nevertheless, there you go. And there's other things that can be done with this. So CRISPR is considered a breakthrough in biotechnology because its ability to target and cut DNA precisely where it needs to go, and then replace the defective genes by doing that. And so anyhow, I we'll talk more about this at a later time, but you need to know about CRISPR because that's going to really change the future. The risk of CRISPR: there's legal issues surrounding who invented it. Well, of course, we know who invented it, and they patented the technology, and so they had a lot of issues. And that patent Patents are a legal process by which you can cover a certain technology. If somebody uses it, they have to pay a royalty for it, and which is good. I mean, it actually helps build businesses, so it protects it so that you can invest lots of money to develop and produce things using that technology. And so it comes down to who owns it, what can you patent now, the ethical issues for that, like for editing human embryos and having superhumans and all sorts of issues about that. So anyhow, I just wanted to bring this up. We will talk about this again later on. So CRISPR is a DNA editing system. It's very precise and very efficient. It's considered a significant breakthrough because of the ability to pick out exactly the DNA that we want. So genetic engineering is taken now to a higher level where we can do gene therapy. And instead of patching or giving pharmaceuticals to treat a disease, we can cure it. Biotechnology can improve food with genetic engineering and recombinant DNA technologies. Naturally breeding corn, we've been able to do that. This is what corn originally looked like, and then through selective breeding over the generations and generations, this is to what we've come to know about it. And so this is sort of the natural way of doing things, but we can circumvent those many years by using manipulations and genetic engineering. So we can speed up the process to make changes. And one of the changes we did with this was golden rice. To make golden rice, beta carotene produces genes that are obtained from other species. So we took the beta carotene. Now, of course, you know, beta carotene is one of those precursors that we use in our bodies use for vision. Let's say if you want to improve your night vision, you can take beta carotene or vitamin A, and you'll improve your night vision, as an example. And so we can clone these genes from the daffodil, insert it into the bacteria, and then insert from the bacteria into the rice. And now we have the rice expressing the beta carotene characteristic that white rice didn't have, and now we're adding more nutrient viability to that rice, and this changes the vitamin A content 25-fold. And so this is just one example of trying to deal with the world population and vitamin A deficiency. So we see this happening now. Corn, cotton, and soybeans, genetically modified. Two factors driving the adoption of modified crops of plants with insecticides engineered into them so they can kill what normally eats it up. Plants with herbicide resistant genes engineered into them so you can spray them with like Roundup and things like that, and then the plant is resistant where the weeds aren't. And there's a lot of controversy over that. But just explaining what the technologies are. And I'll give you a chance to review these in a paper I have about GMO, and then you can take a position on that. Insect resistance is one I was just making a reference to. BT corn, that's Bacillus thuringiensis. That's where the BT comes from. This is a microorganism. Kills insects, pests, but does not harm humans. And so if we clone that from the bacteria, put that in a corn cell, and then the parasite tries to eat the corn, it will and digest a little bit, and then the insect no longer is going to be viable, and now we've dealt with that threat, so the plant will do better. So it comes down to financial viability. Or we can spray herbicides. Here's a sort of a helicopter that's remote controlled. Now they're using drones and various things to spray herbicides. It's the herbicide is in this little pallet there, and it's being sprayed out various sprayers. And notice he's not wearing any face mask. It's a herbicide, probably Roundup or something. Uh, that's a flag for me. I just it just makes me feel bad that that's the case. But you can do that with these herbicide resistant plants. So the plants will break down the herbicide, but the weeds can't. So you can see how nicely everything's growing. I doubt that he has the herbicide resistance genes. Not really funny though. Faster growing, bigger bodies. We can put growth hormone genes in fish, make them grow faster. Um, you know, they have to be FDA approved, and there's all sorts of transgenic animals now that we're dealing with. And is it good or bad? I can't say. Uh, I think I try to stay away from GMO foods. It's just my personal opinion. But we'll see what you think. Biotechnology has led to important improvements in agriculture using transgenic plants using biotechnology, and modifying animals too is quite interesting, and what's going to be going on as in your careers. Even more significant is the extent at which biotechnology has reduced the environmental and financial costs of producing foods, which are dealing with the how to feed the world. Right now, we have 7.4 billion on the planet, soon to be 10 billion. We got to do something about that. Now, trying to reduce the population, that's a whole different ball of wax here. But the potential ecological and health risk of such widespread use of transgenic species are not yet fully understood. And in other words, it's going to be your generation is going to figure out the health risk of such things. So that's all I had for this part one. We'll be moving into part two, going into discussing more of the GMO risk, and then I'll sign a paper for you to review after we get that under our belts in part two. So thank you very much for your time, and I hope this was interesting. And feel free to get those links out of this presentation and see if there's any areas in biotechnology and career wise that might interest you.